So anyway, I was telling you that we are focused specifically on these particular compounds, which are the N-acetylethanolamines, which we can consider them as a sort of endocannabinoids or endocannabinoid-like compounds. And their role in pain modulation, specifically in the processing of pain and pain chronification. And today I would like to spend more about the central sequelae which are associated with the chronic pain, particularly with neuropathic pain. So the neural maladaptive modifications, like for example brain plasticity and reorganization of the circuitry, which are associated or related with the altered resting state and pain evocate functional connectivity, as well as the altered structural integrity and connectivity at the level of white matter, and also microglia, astrocyte, and so on. It's not only a neurocentric vision of the idea about the chronification of pain. But as you can see here in this scheme, I will focus specifically on the limbic system, on limbic centers which are actually modified and long-term modified, which are responsible for the the change that the clinicians have observed for many years, or at least we have recognized and accepted also in the DSM-5, for example, in the psychiatric textbook of the United States, Psychiatric American Association, that actually we have that mood anxiety disorder characterize chronic pain situations. So the literature is very, there is a good evidence, medical evidence that this is in fact occurring. Could I have a silence, please? It's too much noise over there. So we have contributed at least 10 years ago in this review with other colleagues in the United States about the, the four brain pain mechanisms that actually are responsible for the affective states, anxiety, depression, hyperfrontality generally, which are responsible also by hypocognition in these animals and patients. So we focus on the animal models as a pharmacologists, or basic pharmacologists, to understand what is actually happening in the brain and outside of it. So neuropathic pain, in the beginning of my career, at least 35 years ago, more or less 35 years ago, when I started in pharmacology department in Naples, together with other colleagues now working in other part of the world, in UK and also in Canada and United States, we have analyzed the possibility that glutamate was playing an important role in that story. And actually this was the case, but that it depends from the, the input that the glutamatergic synapses receive that can release or co-release, depending from the degree and the chronification and intensity, etc., of pain, these challenges, we can have different release of several other compounds together with the glutamate, the same synapses. So there's a co-release of substances, endogenous transmitters, like glutamate together with BDNF, anti-3, ATP, and so on. And many other compounds, like some substance P, for example. So depending from the challenges on the degree of pain, we can have the sort of cocktail and transmitters that cooperate together postsynaptically and as well as presynaptically in a way that we can have a reorganization of synapses. And you can see here in this scheme very clearly how we can have some important change at the level of the anatomical modifications in terms of there is a doubled synapses, is divided into pieces, as well as here we have also the change in terms of the component postsynaptic receptors. So in other words, we can have an important long-term modification. And this is actually what is happening here. You can see that we can observe, for example, that not only there is a co-release, but also at the level of postsynaptic terminals in the dendrite spines, we can have the release locally of many other compounds, like the endocannabinoids, or prostaglandins as well, or prostamides, which altogether are the same, they altogether belongs to the endocannabinoidome, as we know, recognize this system. And actually, you can see here that there is two important phenomena we've described, which is the depolarization-induced suppression of excitation, like this one, the depolarization-induced suppression of the excitation, because this is glutamatergic terminals, And in fact, this is a suppression of excitation because secondly, or thirdly, at the level here, postsynaptic level, we have the release, the synthesis and release of 2-AG, which is an endocannabinoid, which, very ultra-short feedback, negative feedback, inhibit the further release of glutamate. And so in this case, we can have a sort of break for an overexcitation synapses, a self-control. And also there's a control for the inhibition. So the depolarization is suppression of inhibition, in that case, on GABAergic terminals. So you can see that the complexity of this organization, long-term maintained, can definitely be important for the central sequelae, for the psychiatrical component of chronic pain. And as I mentioned before, endocannabinoid belongs to this system, many compounds. And classically, all of you are aware that there is, for example, probably it's better with the mouse here, the anandamide, together with anandamide, many other compounds, which are the N-acetylethanolamides, which is on the title of my presentation. So I will focus specifically on this part here. But also you have to consider that we have to recognize that many other compounds belong to this complex system of the endocannabinoids. So we are in front of a new idea, a new era of pharmacology, because more compounds, endogenous compounds, all endocannabinoids, very different from each other, and different receptors, or very complexity or promiscuity, of receptor on which they can work on. So anandamide and palmitoylethanolamide, so are very similar to each other, but the difference is that this lipidic residue is actually arachidonic acid, arachidonoyl ethanolamide, or anandamide. This is the palmitoyl ethanolamide, so anandamide, similar to anandamide, but there is here, instead of arachidonic acid, palmitic acid. So the difference is not only chemical, but also in terms of physiology, or pharmacology, or dynamics, the pharmacodynamics, because the receptors they occupy and they work on are completely different. So in other words, we have that anandamide is capable to operate on CB1 and CB2 and TRPV1 and some other receptors, but palmitoylethanolamines work mainly, some part on this receptor, but mainly on the PPR-alpha receptor, which is a nuclear modulator for the expression of several genes. So gene expression is affected by several compounds, several endogenous proteins, receptors, acceptor nuclear factors, and this is one of the nuclear factors on which we are aware, we know that in fact PEA is capable to bind it, to work on it, and to stimulate. So, so in other words, we have to consider this mechanism, a basic mechanism, that when PEA is able or is capable of binding or bound to PPAR-alpha, it is able, this receptor, to heterodimer, to form an heterodimer with the ARAXR receptor, so a receptor for the retinoic acid. So this heterodimer translocates in the nuclei, in the nucleus, and that level is capable to modulate the expression of several genes. So more or less the same as cortisones. So this is a sort of a proteocortisone, because on bacteria this system is well preserved, but as well as in human being, of course. But the same is not occurring for cortisone, so in bacteria is not present. So this is very well preserved in terms of phylogenesis. So it's very ancient, very antique, very ancient system. So in this way, we can have this modulation for the activation of several genes, expression of several genes, etc. So what we have discovered in my lab 2-3 years ago, in 2017, we have seen that this system here, the PPAR-alpha and ARAXR, when activated by PEA, is able to bind directly to the promoter of the CB2 receptor. So in other words, we have the upregulation of the CB2 receptor for cannabinoids. And this is the reason because we can consider PEA as an endocannabinoid-like compound, because potentiate in one way only, on one route, on one track only, this system, via the activation, via upregulation of the CB2 subtype receptor, which is typical for the immunitary system. And also is important, of course, if you consider brain and pain, for microglia. And you can see here what we found that on microglia, in fact, it is upregulated. I don't want to make the story very long. If you are interested, you can read the paper in the scientific report, which was published in 2017. So you can see, well, probably it's better if you read the paper. But anyway, I just want to focus on this aspect, that when we increase on this cell, this immunitary cell, as well as on macrophages, mastocytes, and so on, this receptor, CB2, we have an important phenotypic change in the behavior of this cell type, because we can have the possibility to induce this cell to be protectant for the brain. So in other words, when microglia is overexpressed in the CB2 receptor, is capable of modulating to shape, in other words, the synapses functionality in terms of morphology, because we can have phagocytosis of some part of the synaptic, pre- and post-synaptic, via the activation. Good activation is not maladaptive, pro-inflammatory, but is anti-inflammatory and cell or neuroprotectant in this case. Just to make the story very simplistic, of course, because the complexity of the several phenotypes is very huge and very, still very open, the question at the moment. But at any rate, just to make the story very simplistic, very easy, I want to say that, I can say that this is basically what happens, that when CB2 is overexpressed here, we have the modulation, a positive modulation. So, there is a sort of saving or protecting the synapse, or maintaining synapses, unless the synaptic is pathological. Therefore, in that case, we have the phagocytosis. So at the end of the day, what happens is always some protectant activity in the brain. Well, this is, I can skip this slide. It's very technical, just showing that there's an increase of, the migration, the same migration, microglia migration in vitro is facilitated or increased in the presence of 2AG and so on. When we have pre-exposure to the PEA, the PEA, palmitoylethanolamines. So when we have this activation, this PEA activation, also we have overexpression, therefore the movement of the cell is facilitated towards something in a way that can resolve some damaging or some inflammatory situation or damaging situation in the brain. This is the case of in vitro, in primary cells, microglia cells, and this is the case in vivo. So in vivo, we can see that the same story is occurring. In other words, we have an upregulation in these different tissues, which is the spinal cord and cortex, prefrontal cortex, prelimbic, infralimbic cortex, the limbic cortex. And this is important in terms of what is happening when pain is induced, because you can see here that actually, if the animals is pre-treated with palmitoylethanolamines, like this one here, and is compared just with animals that are damaged in a way that they produce, they feel, allodynia, tactile allodynia, you can see that when there is pre-treatment with palmitoylethanolamide, the degree of pain is less intense as compared to the control here, where we just received vehicle, where pain was dissolved. Suggesting that the preemptive pre-treatment with palmitoylethanolamines can increase the CB2 receptor expression, which can guarantee us to feel less pain, to be more protected against pain. And not only that, it's also important that in terms of therapeutic capability or activity in these animals was capable actually of decreasing the pain perception. This is allodynic animals, and after treatment, you have a resolution of this pain, this increase of allodynic situation, allodynic feeling. But as I told you before in the beginning of my talk, I was more interested more recently about the psychiatrical component of chronic pain, neuropathic pain. And you can see here that the working memory was decreased here, was ameliorated. The blue line, the blue column, of course, corresponds to spare nerve injury in neuropathic animals. In this case here, you can see there is a resolution, recovery of the working memory. In this test, the Y-maze test. And retention memory, the novel object cognition, also happened the same. And more importantly, the massive phenomena is that depression-like behavior in these animals, the dry suspension, the immobility of the animals suggesting depression-like behavior, was very importantly resolved. And surprisingly, because there is a continuum in these psychiatric diseases, surprisingly, we do not observe any phenomenon, any resolution, any therapeutical effect for the anxiety disorder. To be more sure about that, we also have applied another technique, another test, just to see whether or not it was a problem of test. But also in this case with the marble barring test, because you remember before was the elevated pulse maze and the marble barring, in both these tests for anxiety or obsessive compulsive disorder-like behaviors, we do not have possibility with this drug to have any sort of resolution. So, in other words, we have important capability to decrease some of the psychiatrical events, some central sequelae, but not for all. Some specificity in terms of therapeutic capability of resolution. Well, another important test we have performed to understand whether or not we can have possibility to use palmitoyl etonolomide for psychiatric component of chronic pain, neuropathic pain, we have used the Morris water maze test, which actually is the sort of pool, the swimming pool, where in which the water is colored, is black, and is hidden the platform here. So the animals has to learn where the platform is located in this pool, because it's under the water, and the water is colored, it's black. So the animal is floating, swimming around there, and to try to find where is the platform. So you can see that at 5 days, 7 days, etc., all the groups learn at the end where the platform is. But you can see in this column here more clearly that, in fact, you can see that, sorry for that, okay, all right. You can see here, this column here, neuropathic animals, spare nerve injury. You can see that as compared to the white column near the control here, this is very significantly decreased. So in other words, this indicates the time the animal spent in the quadrant where the platform was located. Because I forgot to tell you that after this learning, the platform is removed. So we removed the platform, the island, to put the animal try to survive. So the animal is erratically floating around here to try to find the platform. And of course, more time they spend in this platform with the treatment, it means that it is increased, preserved the memory, the short-term memory, and the hippocampus. So in this case, the memory is lost because they spend very few time in this quadrant where we placed the platform. Imagine that this platform has been removed. But if you make the treatment, the animal is treated, and you can see that there is a recovery, better than the control at the end. And of course, all this is abrogated, is eliminated or prevented when PEA is co-injected with the antagonist of PPAR alpha, GW compound. So suggesting that the pharmacological specific phenomenon related to the activation, specific activation of the PPAR alpha receptor, this nuclear receptor. Just to try to convince you this complexity of this talk, this part of the talk. So you see that these sham animals, this is the trace of the navigation of the animals in the bath, the water here. And you see that the sham animals, the spare nerve injury, and the spare nerve injury treated with palmitoyl etonolomide. You can see that they spend all this quadrant more time around here, but it's more concentrated here, suggesting that the animals remember better, in this case, where the platform was located. Okay? So all this is associated, related with the memory in the functionality, the function circuitry in the hippocampus. So for this reason, we are focused on the electrophysiological analysis of what is happening in the entorhinal cortex and the dental gyrus of the hippocampus. So in other words, we have the navigator in the brain through which we can organize where we are and when something is happening and what actually is happening. Space-time, the space and the time and the memory and what we have to memorize. Anything is happening actually here in this part of the brain together with the gray cells in the medial and trianal cortex, lateral and trianal cortex. So we stimulate here in the lateral and trianal cortex and record the activity of the neurons in dental gyrus. So what we have published, I don't know when exactly, but I think a couple of years ago, this paper. No, there's no indication. Do you remember when it's published?
Yeah, last year.
Last year. Okay, sorry, thank you. So what we have is this is a basal situation. The spike we have is an example, of course, of a single experiment. And of course, this is after tetanization. We have a long-term potentiation. We can indicate in terms of amplitude and slope of this spike here. This, in the time, for a couple of hours, we record the tetanus burst stimulation in the lateral and trianal cortex and record the activity of the neurons in the dental gyrus. So neurons is synaptic activity is potentiated for a long term. So long-term potentiation, plasticity, memory. In neuropathic animals, spare nerve injury, we do not have any sort of potentiation after application of tetanus burst stimulation, which is indicated by this line here. Okay? And this is present 7 days up to 30, 40 days after surgery, after the lesion we have done at the level of the sciatic nerve. Consistently with the behavioral phenomenon, the preserve of memory maintenance, to keep memory, you can see here that in this reddish dots here indicate that there is a partial recovery. This is the basal. This more or less the basal here, 200. And we have, in this case, a partial recovery with the palmitoyl-electronamines in neuropathic animals, suggesting that actually we have the possibility to make a direct comparison between what is happening in the hippocampus, this pathway of the hippocampus and trianal cortex hippocampus for the memory, and which happened actually in the awake animals, in the alive animals, in the moving animals. And this actually is maintained the long term, as you can see here in the slide over there. So the rescue action of palmitoyl-electronamide in chronic treatment of this on long-term potentiation is once more, in this case, confirmed. This pharmacological experiments in this slide confirm that PPAR alpha is critically involved in this activity. And in fact, the antagonist of the PPAR alpha receptor, which is the GW compounds here, prevented the long-term potentiation here. You see here, there's no possibility anymore to increase or recover as before the plasticity, which is suggestive of the reorganization of the circuitry and the entorhinal cortex, particularly in the dental gyrus and CA1 of the hippocampus, cornus of Ammone. And this is, of course, also confirmed in my lab, cooperation with Vincenzo Di Marzo in Naples, and now he's in Canada, in Laval University. So we have confirmed this, that we have the postsynaptic density, glutamatergic synapses in the dental gyrus of the hippocampus, where there is a recovery of plasticity. We also have the recovery of the postsynaptic density. This blackish postsynaptic component here is where receptors for glutamate are very densely located. So we have the recovery of receptors, as also Western blotting is shown here. You see that the AMPA, this is subunit, important subunit for AMPA, is decreased in neuropathic animals consistently with the weakening of the synapses. The functionality is very dramatically decreased, and memory, and therefore we have depression, of course, also in this case here. As you probably remember from that slide where I showed some behaviors where depression was very important in this case, and depression was resolved also with this treatment with palmitoyl-electronamide. And this is very importantly consistent also with other biomolecular data with the group of Professor UCL. We demonstrated actually in this paper here that BDNF was dramatically decreased in SNI, the neuropathic animals. The triplicate, you see here, that there is a decrease, but a recovery when PEA was applied. Okay, when PEA is applied, we have a recovery as the control, as the sham animals. Consistently with this decrease, we have depression, and consistently with the recovery of the BDNF, we have a resolution of depression. And also, accordingly, we have an upregulation as a compensatory mechanism of TrkB receptor, which is in the main receptor for the BDNF. We have also here normalization of the upregulation, this important receptor on the surface of the postsynaptic terminals. So, in conclusion, we can say that chronic pain is associated with long-term potentiation and memory deficit, which is also important in terms of to explain the decrease of the mood, depression. So this is very typical for patients suffering from chronic pain, neuropathic pain. Important is the role played by this endocannabinoidoma, glutamate, GABA, all the BDNF, of course, neurotrophic factors. Remember that all the antidepressant drugs are capable of increasing at the end the BDNF level in the hippocampus, and we can have this with these compounds. So it prevents the spinal injury-associated long-term potentiation change, decrease, impairment, through the activation of this nuclear factor. We have the modulation of the synaptic activity of glutamate, GABA, recovery some important proteins for the construction and rebuilding of the terminals in the brain, the synaptic terminals. So the idea is that to try to treat pain with these compounds, not only to resolve or decrease allodynia and the hyperalgesia, but also important for the central sequelae, like depression, anxiety, and so on, which are associated with chronic pain. Of course, I have to thank all people who work in my lab and also the other colleagues, not shown in this slide, in other part of the world. And of course, I thank you, few survivors in this room, for attention. Thank you so much.